Till we sort out this small technical issue. It's okay? Okay, I think we can proceed. Perfect.
Okay.
Just waiting for it to appear.
Okay, thanks for the kind introduction, and thanks for the invitation. It's a great honor for me to be here and present our work today. I actually changed my presentation after the excellent talk given this morning regarding the same subject, multiomics. So I'm an analytical chemist. I've been working on clinical proteomics for various diseases. So I will mainly talk about some aspects of proteomics and glycomics more specifically, and some research about our previous work on ovarian cancer, and I will discuss some facts and some difficulties related to omics studies. So the omics is one of the, so there is some sort of a mess up in the slides. I hope it's not going to affect the following ones. But omics in general is one of the most commonly used suffix in the last couple years. It basically refers to the comprehensive study of research, whatever your target is, whether it's a protein, whether it's the gene or metabolite. So as you're all aware, so developments in the genomics has changed our understanding about various diseases, and it just introduced new technologies that we can use to treat and understand the biology of specific diseases. But unfortunately, genomics itself is not enough to understand the basic complexity of biological systems. This is why we need to implement more and more OMS to our studies. So whenever we talk about proteomics, we talk about proteins in more specifically their abundances, their localizations, and their interactions in biological system. And this system could be as small as cells or large organisms like humans. So similar to the genomic studies, now there's an important research going on human proteome. We are trying to understand and profile the proteins in our body, in our organs. And it is really interesting that we do see similar profiles in different parts of the body, but the compositions and their concentrations could differ from one part to another. So in biomarker research, when we talk about proteins, we often assume that those are single entities. I like to show this analogy, but I think it's really informative because a single protein can come in different shapes. So the variation that we observe here is related to the proteome, and the proteome involves different parts of proteins, the protein forms. And these forms could be protein-protein complexes, protein-ligand complexes, and more importantly, post-translational modifications. And I would like to take your attention to here, imagining that all these eyebrows, eyes, and mouths are some sort of a modification. Position of this modification could give different impressions. So it's the same thing in the biology. So if the modification is different from one protein to another, they could give different signals. So this is what we observe in post-translational modifications. In fact, there are about hundreds different types of post-translational modifications available. So among them, I'm going to talk about glycosylation. And glycosylation is one of the most abundant post-translational modification. Almost 60% of the proteins in our body are glycosylated. And what these glycans are? Glycans are complex sugar molecules. Here, the structures of those glycans are represented by these cartoons. But if you would like to have a look in detail, it is a complex sugar chain composed of various types of sugars, including galactoses, mannoses, N-acetylgalactosamines, galactosamines, fucose, and sialic acids. And we all know that those sugar molecules play an important role in biological systems. In fact, there are two types of glycosylation, N-glycosylation and O-glycosylation. So glycosylation is taking lots of attention recently due to COVID. We have realized that actually on spike protein, so it's heavily glycosylated, and there are about 22 glycosylation sites. And this is the feature that we observe in here. These purple line represents the spike protein, and the blue ones are actually representing different glycan structures. whenever we look at the publication, you will see the distribution on the glycan composition. As I said, so there are about 22 glycosylation sites, and each glycosylation site we can have different composition of glycans. And when you think about the possibilities of how these glycan compositions could rearrange, you will see the complexity. Actually, this is one of the reasons glycan research has been undeveloped, let's say, because it's very challenging. So it's not easy to sequence the glycans as you do normally with proteins or the genes. So you need to consider the structural isomers, you need to consider the linkage isomers, which they play important role in biological systems. Actually, it's not the first time this glycosylation is becoming popular. So it's an old movie. It's about another rare disease, Pompe diseases. And if you give a close look to the board, you will see the same structures of glycans. And this particular rare disease is associated with a deficiency of an enzyme that involves in glycosylation pattern. And then they were trying to apply an enzyme therapy in order to cure these patients to improve their lives. Glycomics and cancer is highly associated, and there are two main aspects of this study. The first one is the development of the progression of a cancer can be monitored through glycans, glycosylation. And the treatments, most of the monoclonal antibodies that are commonly used right now are actually glycosylated proteins. I'll come to that later on. So in my research, I was lucky to work on various different cancer types: prostate cancer, gastric cancer, breast and ovarian cancer. And when we profile these glycans from serum, we realized that there are a huge overlap between some cancer types. For example, high mannose types of glycans were found in almost all cancers. So those type of cancers are glycans are considered as an immature glycans. That means it's just at the beginning of the glycosylation cycle, and it's clearly emphasizing that the cancer is related with glycosylation. And we do observe some specific glycans associated with specific cancer types. So we have done an ovarian cancer study that we got samples from gynecological oncology group back in Davis, UC Davis Medical School. And we had control samples from females. We have the low malignant potential tumors, and we had epithelial ovarian cancer samples. And we did an extensive glycan profiling. And what extensive glycan profiling is, we don't only monitor and identify the glycans, we also separate the isomers. So it's this isomer-specific glycan composition. And then isomers specifically, if you just go into the details, all these peak represents specific glycans. And even if you go into the deep, you will see that we will be able to identify different isomers, even at very low concentration levels. And after all, we find out that there are specific glycans are highly associated with ovarian cancer. For this type of glycan is considered as a complex type glycan. And what's important in here, we do see sialic acid and fucose on this structure. And we know that those sugars are highly related to the immune system. And what's more important, that within the same composition of a glycan, we can have 5 different isomers. And this isomer separation gives you an important clue, because not all the isomers shows the same efficacy when we, the same specificity when we identify the biomarkers. One of them shows p-values 10 to minus 8, the other one is barely significant. And this is basically telling us that actually the structure of a glycan can affect the biological outcome. And this is another example. So we did find this particle specific glycan. When you look at the overall p-value, you will realize that actually it's not really significant. But when you separate the isomer, you will see that the first isomer is the one actually differentiating ovarian cancer from the others. And then we did another study to verify our findings. We had the training set and we had the test set on a similar cohort. And what we have done later on, and we compared the CA125 results, and I would like to emphasize the fact that CA125 was not significant between those groups, but using these glycans, we will be able to separate the groups using a single marker-based strategy. And when you apply a multimodal approach, you will see that we observed actually the 11 glycan marker panel give us the highest accuracy and specificity to separate disease groups. So the glycomics is not only related to cancers in terms of biology, but also related to therapeutic antibodies, because those are mainly IgG proteins, and those proteins are highly glycosylated. Actually, I was very impressed when I see the talks over these 3 days that majority of the treatments were based on bevacizumab. This is one of the antibody that I characterized earlier to see the glycosylation profile. So the even if they all IgG based, and production of these antibodies are not the same. Some of them are human, some of them are chimeric, some of them are from mouse. Based on the environment that they have been grown, the glycosylation can heavily change, and this is why we often observe batch-to-batch variations in monoclonal antibodies. And in fact, it has to be further investigated how this could affect the treatment. I will also give some important information about how we apply proteomics or glycomics in clinical samples. In fact, we can use any biological samples from cells to liquid biopsies these days, becoming really popular. And what we do is we extract the proteins from the samples, we digest them into small peptides, and we use only specific peptides to identify the proteins. And we also do a second level of characterization to make sure that structure is unique and specific. So clinical applications of the omics study involves in various fields. Personalized medicine, drug response prediction, study of new drug targets, host-pathogen interaction, as we have seen in the COVID case, and more importantly, in biomarker research. And we are all here to find better and more accurate biomarkers. An idea is to predict the diseases or understand their mechanisms to improve patients' life. And the idea here is to bring the research in the lab to the bedside in order to have, again, more accessible biomarkers. So there is something going on in the presentation. I would like to apologize for this. I don't know what happens. Probably there is a characterization. Yes, don't worry, it's not an important thing. But I would like to just show you the basic biomarker development pipeline. How it works, first of all, the first phase is the discovery phase. And we got lots of samples. We try to profile everything we can do. And then the next step is to verify them and then later on validate them in separate cohorts. But what we do see is, unfortunately, is the case, developments of in the omics technologies change our world. We have a number of publications, we have a number of research, but unfortunately we don't see too many new markers. And even if there's new markers, the specificity and the sensitivities are not really high. And whenever we would like to just go back and try to understand what is the root cause of this effect, is that majority of the markers that has been discovered in the first phase, unfortunately, cannot be validated in the second cohort. And then there are obviously lots of reasons behind that, and poor experimental design, even sampling, storage, variation between the clinics, even transferring the sample from one site to another could affect the results. And we do see that, especially in the omics technologies, there is no standard protocols. So everyone is using something similar to another, but unless you apply a validated protocol, there will be always variations. And, okay. So here I would like to also talk about the omics era. As I said, now the technologies are becoming accessible. We have those fancy instrumentations almost everywhere in the labs, and then we can generate big data. And because assumption is if you generate big data using proper statistics, we will be able to solve everything. Normally, with omics technologies, we were able to analyze more and more samples. We would be able to get more and more feature in less time and less code. But in fact, we are just missing an important information, because in general, what we are looking for is the biological variation between the groups. But often this biological variation is masked under individual variations because we cannot really phenotype the diseases. We just pull controls from one cohort to another. And then there is a huge person-to-person variation that we cannot actually get the real biological variation that we hope to observe in biological system. And what is also very important that we observe a huge technical variation between the measurement. And unless we reduce them, we wouldn't be able to find or verify any new markers. And then we have to be careful when we talk about big data. Everyone is obsessed about a big data. So I'm going to give you an example about a single study. Using the same samples, just changing the protocol from one to another, you can increase the number to 100 to 200. By using a difference instrumentation, you can increase this number to 200 to 500. By just relaxing the search parameters, you can increase this number to 500 to 1,000. So if you're only looking for the numbers, obviously you would like to hit the highest number. But what we're trying to do is to do and measure everything reproducibly. So we need to find the balance between the technology that we apply, and we need to be aware of these drawbacks actually that is commonly seen in omics research. Another important point is actually when we talk about different omics, genomics, proteomics, metabolite, and glycan, the first thing that has been developed obviously among them is genomics. So we had number of data available. Then development technologies enabled proteomics, metabolomics, and other omics to grow. But when we talk about a data analysis perspective, we see that one size doesn't fit for us. We know how to treat a genomics data, but if you would like to apply it for the others, it may not be suitable, because whenever we look at the genomics data, we have one-dimensional information. We have the sequence or we have the modification, and the outcome is just presence and absence. So you don't have too much of information. So it's easy to predict the differences between the system. But whenever we look at the other omics, we see that we get a multidimensional data. We often get IDs, whatever that particular compound is, but we often neglect modifications, as we mentioned that earlier. The outcome is not presence or absence anymore, because absence is considered as a missing value in majority of the statistical calculations. And then it brings another question in the clinical research, because maybe we are missing a very important and valuable information. And where we stand among those as an analytical chemist is to be a bridge between the clinicians and the statisticians. So we would like to develop new technologies. We would like to validate the platforms. We would like to apply quality control mechanisms to every single step to get a reliable and reproducible information. And the way to do is, again, using standardized protocols, do the detailed phenotypings, try to understand the biology behind it. When it comes to rare diseases, it's really difficult because you cannot have these large N numbers anymore, so it's very difficult to find the patient and also find funding for this particular diseases. For rare diseases, generally, a multi-omics-based approach is considered in order to get as much information as possible from an individual. This has been covered in the morning, as I said, so I'm not going to go into too much in detail. But combining omics technologies will have lots of benefits, because most of the time the biological systems or the diseases that we're interested in involve more than one particular biomolecules. Maybe it's metabolite plus proteins, gene plus protein. So we need to look at the question from different perspective. But the limitation that we observe in here is most of the time, unfortunately, we don't have the technology to evaluate the data yet. So there is no, there are new, let's say, tools available right now in order to help us to evaluate the outcome, but it is really lacking at the moment. So it's very difficult to align this different dimensional data in one scale. And the future is to use standardized protocols to have controlled assays, design it from the beginning to make sure that you will get some sort of a reassurance in your measurements, and then collaborate different fields, including chemists, biologists, medical doctors, obviously, and the statisticians. And sometimes it's good to look out of the box. As I said, glycomics is there, it's hugely abundant, but it is neglected, so it might be a new way to consider, I believe. So it doesn't look nice, but I would like to just mention about a new project has been recently funded. So it's in collaboration with İnönü University and Yüksek İstisas University that we are going to look at polycystic ovary syndrome samples and the endometrium cancer samples, and we will also include some rare endometrium cancers into our study. So it's going to be a targeted proteomic study. Hopefully in the future meetings I will be able to present some results. And this is our team. So we are mainly working on proteomics, global protein profiling, targeted protein profiling, some structural proteomics to identify the complex proteins and protein complexes and so on. And finally, this is our campus. We are in Ankara, the capital of Turkey. It's a governmental city. I know that it's not very appealing, but at least our campus is sitting in a huge forest. It's about 45,000 meters square. And then I hope you will enjoy if you ever come and visit us. And I would like to thank again for your time, and I'm happy to take your questions.
Truly been a very, very interesting presentation, and I really like the graphics. And I doubt I've ever been at a more interesting multi-omics lecture, because I always find it, as a clinician, I always find it a bit challenging to understand all these new advances. But you've really made it look so simple and so clear. So thank you. Are there any questions? Yes, Professor Grigor.
I just wanted to ask about these new projects you plan to do. And one is polycystic ovary syndrome, and it's also associated with glycolized
No,
proteins.
that particular one, the project itself is just targeted proteomics. We have set up a panel of proteins that we would like to quantify, but we will do an extensive quantification. This particular project doesn't involve glycomics, but we are thinking about applying new projects. It's all about funding. Unfortunately, we cannot do all at once.
And the endometrial part is for also younger women in reproductive age, or it's for...
No, it's a younger woman, so it's about between 25 to 40.
Okay, so we can discuss that later.
Perfect, thank you.
In fact, unfortunately I couldn't remain here, but I am aware that even the Bulgarian colleagues, they have been looking into endometrial cancer in young women. And it would be interesting to see how many of their patients actually had polycystic ovaries, because we know it's an association. So this could be definitely room for collaboration with different members. I know that Irina, of course, is interested in endometrial cancer. There was Professor Ristok, Professor Giordano. So for sure, if you need international partners.
So actually, our collaborators, Professor Kerr, might be on the audience. I'm not quite sure whether he's there.
I know he registered as a participant.
Yes, he did.
So,
yes, if he's in the audience, he might be able to give you more details about the cohort and the data or sample availability.
I will look for him.
But it might be a little bit late for him as well. Anyways, if you have any questions or if you would like to be in contact, I'm happy to arrange it.
unfortunately, I can't see him, but we'll definitely be in touch because we were in contact by email, and I know that is part of cost, so I can't see him there. But no worries. But thank you so much, and we look forward to hear more about your findings.
Thank you.
Thank you so much again. Okay. So now we move on to our next distinguished guest. It's Professor Kubel Kaketi, okay, who is from the University of North Macedonia, and she's going to be speaking to us about rare ovarian cancers. So we look forward to your presentation.
Good day or nearly good evening to everybody. It is my honor to be part of this special conference. I consider myself a lucky person in general, but today to give a lecture after this excellent lecture on such a specific and difficult topic, which was given to us in such simple manner, I think I'm not lucky. My presentation is about rare ovarian cancers. I decided that the rarest of them all are malignant germ cell tumors, so the presentation will be about our experience in malignant germ cell tumors, and we will present few cases. I work in the clinical hospital Acıbadem Sistina. Is it okay? I work in a clinical hospital Acıbadem Sistina, which is part of a larger hospital groups of Acıbadem. It is located at the river bank of the beautiful river Vardar. I don't have the information from the national registry, which I don't think even exists on the incidence of ovarian tumors. But in the period from 2014 till 2022, 619 ovarian neoplasms were diagnosed in our department, of which 213 were primary ovarian malignant neoplasms, and 45 were secondary or neoplasms metastatic to the ovary. Of course, the malignant germ cell tumors are rare ovarian malignancies, but benign counterparts, which is benign cystic teratoma or called dermoid cyst, is not so rare. This is more common tumor, which occurs usually in women during their reproductive years from teens to 40s. Germ cell ovarian tumors develop from, as the name says, from germ cells. Malignant ovarian germ cell tumors are divided in several groups: immature teratomas, dysgerminomas, yolk sac tumors, embryonal carcinoma, non-gestational choriocarcinoma, and mixed germ cell tumors of the ovary. I will present 3 cases of different types of germ cell tumors that were diagnosed in our department. First one is a case of dysgerminoma. First, a few facts about dysgerminoma. It's a primitive germ cell tumor which is composed of cells that don't show any specific type of differentiation. This is the most common of all malignant germ cell tumors and accounts for about 1% of all ovarian malignancies. It arises in children, in young adults, and sometimes in phenotypically female individuals, but which have gonadal dysgenesis. Usually the patients present with abdominal pain, swelling, and elevated serum levels of LDH. Rarely, serum levels of HCG can also be elevated, but in this case, usually we as pathologists find scattered syncytial trophoblastic giant cells in the tumor. Patients may also have paraneoplastic hypercalcemia, which is good information is that most of the patients present with stage 1 disease. Bilateral involvement is grossly evident in 10% of the patients, and we do not expect to see elevated serum levels of alpha-fetoprotein in patients with pure dysgerminoma. As most of the germ cell tumors, this germinoma also has chromosome 12 abnormalities cytogenetically, typically isochromosome 12p or 12p amplification, which is seen in 80% of these germinomas. Also, KIT mutations are present in around 50% of these germinomas, and the amplification of KIT in 30%. In patients who have gonadal dysgenesis, this germinoma may present in association with gonadoblastoma. As most of the germ cell tumors, these tumors are large, usually around 15 centimeters in diameter. They're fleshy, yellow or cream-colored, usually lobulated. They may have cystic areas, hemorrhage or necrosis. On microscopic examination, about 20% are bilateral, but this can not always be seen on gross examination. The essential pathologic diagnostic criteria include finding of uniform rounded primitive germ cells with clear or eosinophilic cytoplasm, as seen on this slide, with presence of macronucleoli. Just a second. These cells are arranged in nests, cords, and trabeculae, and they're separated characteristically with fibrous septae containing lymphocytes, which are most often T lymphocytes. It's desirable in germ cell tumors always to perform immunohistochemistry to be sure that we're not missing any type of differentiation. So for this germinoma, it's desirable to have positive stains for OCT4, SAL4, KIT, and or D240, which is podoplanin. These tumors, as all malignant tumors, are staged according to the TNM and FIGO classification system. The prognosis depends on the stage of the disease. 10-year progression-free survival is luckily more than 90%, and recurrences, which usually happen in the first 2 years of initial presentations. Initial presentation can be seen in around 10% of the patients. Of course, stage is the most important prognostic factor. Our case was a patient, 25 years of age, who was pregnant. As the literature states, the most common tumors malignant in pregnancy are these germinomas. So this patient had this germinoma. She had slightly elevated levels of CA125 and alpha-fetoprotein in the serum, but this elevated alpha-fetoprotein is within normal range for the pregnancy, for the month of pregnancy that she was in. Other tumor markers were within normal range. The ultrasound showed oval tumor formation behind uterus, which on ultrasound measured around 75 millimeters. Right adnexectomy was performed, and the ovarian tumor actually measured 170 millimeters, and residual ovarian tissue was seen on the surface of the tumor, which measured maximum 40 millimeters. Tumor surface was smooth, gray-white with the iatrogenic tumor rupture. Unfortunately, I don't have the macroscopic images. The cut surface showed homogeneous gray-white tumor with alternating softer and more compact tumor areas with some foci of hemorrhage and necrosis. There are some microphotographs showing nests, sheets, and trabeculae of monomorphous tumor cells separated by fibrous septae containing lymphocytes. Tumor cells are polygonal, well-defined borders, one central nucleus with one or two prominent nucleoli, and mitosis were common. It's rare to see tumor cells arranged in cords, which can sometimes be confusing, but it's part of this germinoma pattern. And also there can be areas of necrosis as seen in the second slide. The immunohistochemistry showed positivity in the tumor cells for PLAP, KIT, and D240 or podoplanin, as seen here on these microphotographs. This staining pattern is membranous, is characteristic of this germinoma. Negative stains were observed for cytokeratins, common leukocytic antigen, CD30, alpha-fetoprotein, HCG, inhibin, WT1, SOX10, and vimentin. Some of them are given here in microphotographs. We can see high Ki67 proliferation index and also confirm the presence of lymphocytes in the fibrous septa. 4 months later, cesarean section was performed. She gave birth to a live baby. Biopsies were taken from the left ovary and omentum. No residual tumor was found, but after that we don't have any information about the follow-up of this patient. The second case I'd like to present today is a yolk sac tumor. Yolk sac tumor is a primitive germ cell tumor which displays multiple patterns of differentiation towards endodermal tissue. It can be towards extraembryonic endodermal tissue or somatic tissues, like secondary yolk sac or allantois, or if it's somatic tissues, then we can observe intestine, liver, or mesenchyme. This tumor accounts for 20% of malignant ovarian germ cell tumors. It usually appears in the second or third decade of life. The presenting symptoms are similar to this germinoma. It presents with abdominal pain or pelvic mass. But here, the patients usually have elevated serum levels of alpha-fetoprotein. What is interesting that yolk sac tumor can also be found in older women, but usually in association with endometrial or ovarian epithelial cancer, in which case it is considered to be a somatic variant of the tumor, and in that cases, usually it does not have cytogenetic abnormalities on the chromosome 12, which is most common again, isochromosome 12p. Tumors are usually unilateral, large, again around 15 centimeters in maximal diameter. Macroscopic examination is similar to this germinoma. They can be solid with cystic areas, friable, hemorrhagic, and sometimes necrotic in appearance. But when we say hemorrhage and necrosis for a germ cell tumor, we must also be prudent to exclude possibility of choriocarcinoma, which usually has areas of hemorrhage and necrosis. Essential diagnostic criteria for pathologists are finding of primitive germ cell tumors exhibiting endodermal, extraembryonic, or somatic patterns. It is again desirable to perform immunohistochemistry, which shows alpha-fetoprotein positivity, which on the other hand can be only focal, and also positivity for glypican-3. Staging is again as in other malignant tumors according to the TNM and FIGO classification. The 5-year survival rates are more than 95% for early diagnosed disease in stage 1 and 2, but it falls to 50% for stage 4 of the disease. If yolk sac tumor is found in older women associated with epithelial cancer, ovarian or endometrial, then it has worse prognosis. Our case is a 25-year-old female who had nausea, abdominal pain, and bloating. For these symptoms, CT scan was performed, and tumor was found in the right ovary, measuring 170 millimeters with cystic areas. Right adnexectomy was performed with partial resection of the left ovary, appendectomy, and omental biopsy. On gross examination, the tumor measured 170 millimeters with smooth gray-white surface. The cut surface of the tumor showed mainly solid and partially cystic, friable, sometimes dark yellow areas with necrosis, and there were few cysts with maximal diameter of 30 millimeters. The histology showed a reticular microcystic pattern, rarely endodermal sinus pattern with characteristic Schiller-Duval bodies. Focally, there was a glandular pattern with intestinal type glands. Tumor cells exhibit variable atypia, clear cytoplasm, and finding of hyaline globules, which is characteristic for yolk sac tumor. Here there are a few microphotographs of the tumor, which shows in the middle area of one tubular formation lined by single layer of cells with large nuclei and clear cytoplasm. On the image on the right, there are few glands with intestinal differentiation, clear mucin production, and on the left side, there are structures which resemble Schiller-Duval bodies. The immunohistochemistry in this case showed positivity for alpha-fetoprotein in about 20% of the tumor cells, whereas in the area of intestinal differentiation, there was positivity for CDX2 and carcinoembryonic antigen. Negative stains were found for cytokeratins, Paxate, CD30, neuron-specific enolase, napsin, and HCG. We performed napsin here because of the clear cytoplasm, and this stain is usually positive in clear cell tumors, so it's one of the ways to exclude different types of epithelial differentiation. And the third case belongs to the category of mixed germ cell tumors. Mixed germ cell tumors are in fact germ cell tumors which contain 2 or more germ cell tumor components. Usually there are 2, but sometimes there are more than 3 components. Again, these tumors occur in children and young adults. If they occur in children prior to menarche, there can be precocious pseudopuberty. Serum levels can be elevated for LDH and HCG, but also AFP, depending on the time of the germ cell tumor component that is present in these mixed germ cell tumors. Patients usually present with abdominal pain, abdominal mass, or menstrual disorder. Again, many tumors cytogenetically show abnormalities in the 12th chromosome. On gross examination, tumors are again large, averaging 15 centimeters with solid and cystic areas. The appearance depends on the elements that are present, and what we mentioned before, not to repeat myself, the gross examination will show areas that may resemble yolk sac tumors or dysgerminoma, and if there is a component of teratoma, sometimes the tissue can be gritty, which suggests presence of cartilage or bone. Essential diagnostic criteria for pathologists are finding of 2 or more malignant germ cell tumor components. Staging, of course, is TNM and FIGO classification, and the most important prognostic factor is again the stage. It is important for the pathologist to clearly define the percentage of different tumor types present in these mixed germ cell tumors, because the chemosensitivity for the different tumor types is different. It is not yet defined whether a pathologist should say that the tumor is malignant mixed germ cell tumor. It's if, for example, the patient has immature teratoma with small foci of embryonal carcinoma or yolk sac tumor. If these foci are less than 3 millimeters in diameter, then it is possible to classify the tumor only as immature teratoma, but if these foci are larger than 3 millimeters, then in the current literature it is recommended that these tumors should be classified in the category of mixed germ cell tumors. The most common combination is dysgerminoma and yolk sac tumors, the 2 components that we previously discussed in the 2 cases. But there may be components of embryonal carcinoma, choriocarcinoma, or immature teratoma. Different components of the tumor are usually intimately admixed, but sometimes they can be in the separate areas of the tumor. Our case is 14-year-old female with nausea and abdominal pain. The CT scan showed ovarian tumor mass measuring 165 millimeters in the left ovary, for which left annexectomy was performed. The tumor was lobulated, measuring 170 millimeters with smooth gray surface. Cut surface showed lobulated gray-white or yellowish solid tumor tissue with areas of necrosis and hemorrhage again, as well as small cysts that measured a maximum 45 millimeters in diameter with smooth inner surface. The histopathology showed that this mixed germ cell tumor composed of yolk sac tumor and immature teratoma. 80% of the tumor was yolk sac tumor component with the characteristic pattern that was previously described, with focal area or areas again of glandular pattern, intestinal differentiation. Cells exhibited variable atypia, clear cytoplasm, and findings of rare hyaline globules, whereas 20% of the tumors was an immature teratoma. In the immature teratoma component, there were tubular or cystic structures with tall columnar respiratory type of epithelium, but also there were cysts lined by squamous epithelium and also areas of intestinal differentiation. In 2 sections taken from the tumor, the immature neuroectodermal elements were seen with cellular glial tissue and rare neuroepithelial tubules and cysts, which were found on more than 3 low-power fields. Few cysts were covered with heavily dark pigmented cuboidal cells lining, and also there was immature mesenchymal tissue with chondroid and bone differentiation. In the yolk sac tumor component, we observed positivity, which was focal in 35% of the tumor cells for alpha-fetoprotein, as well as for CDX2 in the intestinal type pattern. Negative stains were observed for cytokeratin CD30, ER, PR, inhibin, and HCG. In the immature teratoma component, it was important to show that there is immature neural tissue, which is the most important in the grading of immature teratoma. So the important immunohistochemical stains here were S100 and glial fibrillary acidic protein. Here are some microphotographs with H&E stain of the both tumor components. On the left side of the slide, in the lower part, there is yolk sac tumor. In the upper part, there are some glands and immature chondroid tissue and mesenchyme. In the middle section, there is this cyst lined by heavily pigmented cuboidal or columnar epithelium. And on the right side, at the bottom, we see on one side yolk sac tumor and on the other side intestinal type epithelium, whereas on the top right side, we can see some glands lined by cylindrical epithelium and also neural immature tissue. The markers for neural differentiation were positive in this immature neural tissue, but also on the top right image, we can see positivity for S100 in the immature chondroid tissue. In this period, in our department, there were also additional rare germ cell tumors that are worth mentioning. One was a consultation case, 6-year-old child with tumor of the left ovary, infiltration of the sigmoid colon. The tumor was initially diagnosed and confirmed as a yolk sac tumor. And another patient was 70-year-old female with abdominal, who presented in our hospital with recurrent immature teratoma with peritoneal gliomatosis, which was previously diagnosed in a different institution. I hope this was helpful to see some insights in our rare ovarian tumors, and thank you for your attention.
This was truly comprehensive, and it goes to show the range of ages. Like yesterday, as Prof. Grigor can say, we had a press conference with the media in North Macedonia and Skopje, and like one of the questions was like, how young could a girl present with a gynae tumor? And as we can see, I mean, especially the rare tumors, unfortunately, do occur in very young women and young girls, like 14, you know, I mean, and younger. So again, this stresses the importance of early diagnosis. I mean, especially now that we're celebrating World Go Day. Are there any questions? Yes, there is one question from Professor Grigor.
Do you have maybe a follow-up of these operated women? Because the first one was lost for follow-up, but the other...
Unfortunately not.
You don't have, thank you.
Yeah, that is the sad thing about following up patients. I mean, I guess they would have had so much of the hospital that then...
Yes,
Yes.
yes, usually our hospital, Acıbadem Sistina, is a private hospital. So very often patients would come for a surgery and pay for the surgery, and then they choose oncologic treatment elsewhere in the public healthcare services because it's so expensive, and and we don't have such a good connection to have the same system and retrieve the data. We have to ask on the phone, but unfortunately for these patients it was not possible.
Yeah, and again, that stresses, it ties in with the previous lectures we've had before on the importance of having a homogeneous register where you have the data, because the data is there. Ultimately, data is helping the patient because you can follow, you can see whether the treatment is working, etc. So really thank you, because this highlights and rips up our ethos, the mission statement of Gynocare. Thank you so much. So now we can move to the very last lecture of this 3-day intensive training school. But just to leave you, with, I can't say with a dream, because it's actually now very real. We're going to end this training school by linking to NASA. Yes, so actually we've got a talk by Professor Afshin Behetchi. Unfortunately, he can't be present here because he's obviously in NASA in the USA, and because of the time difference, it was a bit impossible to connect. So we're going to have a pre-recorded lecture, but I really invite you to follow, because it's really very mind-blowing research, which is the future, well, now and the very near future. So we can...
Hi, my name is Afshin Beheshti. I'm currently a principal investigator at NASA, KBR at NASA Ames Research Center, visiting researcher at Broad Institute, and also president of a nonprofit called COVID-19 International Research Team. Today, I'm going to be talking about, well, first, I'd like to thank the organizers for giving me the opportunity to speak to everyone here. And today, I'll be talking about the topic about the space travel or the space environment, increase the risk for gynecological cancers. So before I go on, I want to give just a brief background of what kind of experiments there are with spaceflight. I'm pretty sure that people not everyone will be familiar with these type of experiments. So why should we care about space biology and space research? Because, some people might think, oh, there's only a few astronauts going up there, so why should we invest all this money when there's more immediate issues, clinical issues affecting hundreds and thousands of people on Earth? Well, one thing is, future explorations, it's always captures the imagination of everyone, what's out there. The explorer, just pure science-wise, what is more than the Earth, and is there any life signs outside of Earth? So, going to the moon, for example, is the next mission. You've heard about the Artemis missions about the launch, and that's going to be further, helping to go further in space exploration, and then eventually, maybe in the next 10 years, we'll go to Mars. And after that, the whole Star Trek scenario, exploring the unknown and the universe. And what's interesting about all this is that, not only does this benefit, let's say, the few astronauts going up there and just people who are space enthusiasts, but also the technology and the healthcare benefits that we create from space biology research will also benefit people on Earth. There has been many examples in the past, like prosthetic legs, where and prosthetic body parts have been originally created by technology created by spaceflight. So the space environment. Now, you might not be familiar with it, but there's several hazards that are there that could impact health. Now, if you look in this right panel here, from this review we had put out in Cell a couple of years ago, there are things like distance from Earth, or confinement, or the hostile closed environment. Those are all concerns that could impact health. The two things that I'm going to focus on is microgravity, or the lack of gravity, and space radiation. And this movie here depicts, one aspect of space radiation is that the solar particle event, so a high acute dose of protons will hit you. That is a little easier to prepare for because you know when a solar flare will happen, so then astronauts can prepare properly for that and maybe take some precautions to reduce the dose they'll get. The other one is after you leave low Earth orbit, and even in low Earth orbit, there's the galactic cosmic ray or deep space radiation. Now, most of the experiments that are done now and all the missions are done now is all at low Earth orbit. So is that the ISS, the International Space Station, or the former shuttle missions. And those were more inside low Earth orbit, which is inside the Earth's magnetosphere, which is here, the Van Allen belt. And there, due to the physics of it, the amount of radiation an astronaut will experience will be a lot lower than in deep space due to, you know, the magnetic field reducing the dose. Now, here's a little chart here to actually see the equivalent doses that you might be able to get. So like an annual, let's say, cosmic ray here on Earth, CO is virtually nothing. Now, let's say if you get a CT scan or, you know, a DOE, a Department of Energy radiation coworker in the US, or even, you know, equivalent in the European countries, will get this high of a dose. Now, a 6-month mission on the ISS is now equivalent here, and let's say 6-month mission going to Mars, now remember going outside of the magnetosphere is this amount of dose. Now, you got to keep in mind something that I'll discuss in this next couple of slides, is that the type of radiation you get in space is very different than the type of radiation we get on Earth, and it's much more damaging. So as I said, this upcoming mission to the Moon and Mars, and of course the microgravity is one of the major other concerns on health, because due to the lack of gravity, there's, you know, many different health risks that could come, and of course the radiation I explained to you in the last slide. Now, you know, if you're a Marvel fan or a comic book fan, you know the Fantastic Four was concerned about this when they went to space and they were bombarded with cosmic rays. It turned them into superheroes, but in the reality, what should have happened was not superheroes, but they probably would end up in the hospital because of all the health risks that would happen to them. So unfortunately, we might have to go back to Marvel and tell them they're not very accurate with their description of what happens when exposed to galactic cosmic rays. But that aside, you know, there's two types of radiation that we experience. One is on Earth, which is, or the gamma radiation we experience on Earth, which is called low LET radiation, which is basically high speed particles going through, you know, right through your cells, what this depicts. And LET refers to linear energy transfers. It's basically, you know, the amount of energy that an ion or a particle would transfer to, you know, a material, or per distance or per cell. So in this case, like, you think of gamma rays as like, you know, sand particles flying through your cells. Now, high LET radiation is what's the heavy ions. Here's a little chart here depicting all the ions in space, from helium or hydrogen particles, protons, to all the way down to like iron particles, the big ones. So what happens with these high LET is that, you know, basically they deposit their lower speed, and then they go into the cell and deposit all their energy in the cell. And this is what causes a lot more damage than that. So this is why a lot of the biology is very different when you compare galactic cosmic rays, which is high LET, to low LET radiation, which is gamma we have here. It's a lot more damaging. So with the microgravity and space radiation, what is all the impact now on health? Now, this is that review we had in Cell a couple of years ago, talking about what kind of risk of different diseases might occur from, you know, low Earth orbit missions, depending on the duration, to deep space, to like even planetary visited habits, like going all the way to Mars for, you know, a year or more, 3 years. So there's, you know, distinct health risks that we know of can occur in either female or male, and then there's sex independent ones too. And overall, there's a lot of different health risks, from cancer risk to muscle, you know, diseases. So that basically there's the muscle wasting or bone wasting, cognitive issues or brain, you know, related diseases, CNS issues, central nervous system, cardiovascular issues. There's even a thing called space associated neuroocular syndrome, where, you know, it packs the eye for some astronauts, could reduce vision. And as you can see, that the longer duration and the more deep space they go, the more risk of all these things are occurred to, you know, for health risks. So that said, you know, how do we mitigate this response, and how do we, you know, understand the biology enough to, you know, figure out how to make it safe to, let's say, go to Mars? And in this talk, you know, I'm going to be talking about the cancer risk mainly. So before I go on, I mean, let's talk about the types of experiment that can be done to, you know, understand this. One is you actually send samples to space. So these are our mighty mouse astronauts. As you can see, these are the mice that are in space. There's some mice floating around, and eventually, the mice seem to acclimate well and like whiz around the cage nicely and seem to have fun. And then there's also, say, if you don't have the grant money to send or money to send samples to space, you could do simulated experiments on Earth. So one place to do it is at Brookhaven National Lab, which is the High Energy Physics Collider in Long Island, New York. And NASA had set up a space radiation laboratory in the early 2000s to actually simulate the galactic cosmic rays, the heavy ions. So you take this high energy physics collider, you could get iron particles, oxygen particles, and things like that. And then, here's the facility where they would then take those ions and, traverse them down here. You can put your samples in front of the beam and radiate whatever you want, cells, mice, and anything else to simulate the galactic cosmic rays. So microgravity simulating experiments in rodents are usually done by, this hind limb unloading, which is kind of a silly thing, but you hang the mice by the tail, and then so it simulates unloading from the back legs. So then that, if you just hang them by for a couple of weeks, is some experiments I had done at Brookhaven National Lab a year or so ago. And then, you get some of this, also the fluid shifts, you might get a little bit of that. So then, after a couple of weeks, definitely the muscle wasting is similar to what happens in space and so on. And then there's also other newer models developed by Sirocco and Mary True, where it's called a partial weight bearing model, where, you could have a harness and the rat could go up and down, simulating different amounts of microgravity events, like so simulate the moon, let's say gravity, or the Mars. Now, there's other types of experiments that I'm not going to talk about today, but you could also do experiments with Drosophila, these fruit flies. People send those to space. People also will look at C. elegans. Each of these animal models will represent some, a specific component of human physiology that you could understand. That's why you use different animal models. Even people send zebrafish, for example. Here's the little aquarium. And of course, people do experiments with plants just to figure out how to grow plants in space, make them self-sustaining, and microbes and cell cultures. And now I just want to quickly say, there's resources that out there that people can use to actually, if you don't have grant money, or if you're just interested to see if your idea will have any legs to walk on to, for future projects, or even if you just wanted to work on something quickly to figure out, generate a hypothesis. There's a resource that NASA offers called NASA Gene Lab, and here's the link up here, genelab.nasa.gov. And this is basically where all the omics ends up, all the transcriptomics, genomics, epigenetics, proteomics, whatever omics under the sun that is done with space biology research around the world ends up in this data repository. And it's not just a data repository, there's ways you can analyze data on there, there's visualization, and then, and they offer the higher order process data, so people don't have to really process the data. They can download that and quickly get some nice results, or look at their favorite genes or proteins of interest and know what occurs in space. We also have these things called analysis working groups, and I lead the multi-omics group. These things were set up initially for determining consensus pipelines, but eventually it turned out scientists around the world want to work on neat space biology questions together. I lead, as I said, the multi-omics systems biology group. We're about 200 something members. And there's a bunch of different projects we're working on, and this is one of the projects that evolved from this. So the majority of the authors in this paper and the future work that's going to come out of this is going to be from this group. So anyone here who's interested, please contact me, and you're more than welcome to be part of the group. The more the merrier, for these projects. And another, if you just want to get a good gauge of what kind of space biology experiments are done, me and Chris Mason, Susan Bailey had put out this large package of papers with Cell Press. There was a bunch of papers we had put out, and then some papers that were, from other people too. So basically it's large, like, I don't know, 30, 40 papers in here, and these are the covers we got. This is Scott Kelly, one of the NASA twins from the NASA twin study. These are the pictures he took on the ISS. But this is a nice resource to get a good gauge of what kind of experiments are done. So now we're going to focus on this paper that came out earlier this year that was led by Jean, your very own Jean Kaliagwas, and from the University of Malta. And these are, majority of these people are AWG members, are from the multi-omics group. So the question that arose, Jean approached me, she said, is there anything known about gynecological cancers during spaceflight? And the answer I gave her, not too many people have been researching this, unfortunately, and it's a very open field here, and it should be studied. And so this is where she led this group to try to start answering the question, what is the risk of gynecological cancer, and so then in female astronauts, and know what maybe potentially to plan for and how to maybe mitigate this response, since it's a very understudied field. So here's the, you know, as I said, this is a very group effort. This was a review we had put out, and now we're working on actually working on like the GeneLab data sets to come up with actual data analysis to figure this idea out. But this review is a good start, and as you can see, the AWG members are outlined by the green box, and there's a lot of AWG members that worked on this, and Gina, as I said, was leading this project and work. So, you know, again, the idea is what is the risk of cancer, and look in the overall literature and reviewing it. And, you know, as I said, there's not that many papers out there on the, you know, how the space environment impacts gynecological cancer, or in general, female reproductive system. So this is a very understudied field, very understudied subject in the field. It's that, you know, really needs to be studied because, you know, we have equal amount of female and male astronauts. And the plan, from what I've heard, is, you know, they want a female to be the first astronaut to step on Mars, you know, so, or end of the moon. So, you know, this is going to be, if you want long duration deep space travel, females really need to understand, you know, the risk of gynecological cancers for them. So in the paper, you know, the first step is like, what is the overall known risk of, let's say, radiation impact on different ages, and also, you know, what is the overall can't know about cancer associated, you know, risk with microgravity and space radiation. So as I said, you know, we know that cancer risk is an issue in space travel, and we know the doses. This is the doses that, you know, the amount of radiation impact they'll get in general. This dose is not equivalent for space radiation, but, you know, in gamma. So this is like the radiation effects on female reproductive system and the damage it might do if you get this, you know, very high doses. So this is in milligrays. So you know, it's 1700 milligrays. You know, this could be high impact, but, you know, when you start getting up to like 20,000 milligrays or 20 gray, you know, that you get some permanent damage on you know, the female reproductive system, and this could potentially lead to cancer risk. Now, remember what I said, that space radiation equivalence is a lot higher damage equivalent. So let's say if for all ages, if they got this, you know, 1700 milligray effect and it's temporary sterility, if this was now an iron particle, which would be like a bowling ball going through your cells and depositing all that energy, it'll be a lot more damaging, and it might be a lot more effective, or it might be ovulatory suppression or something related, a lot more permanent to like these higher doses of gamma. So we know that, you know, there's been a lot of work in the past couple of decades done on different cancers, and they mainly focus the papers that, as has been outlined in their paper here, focused on things like breast cancer. Glioma is one of them. Lung cancer is one of those other ones that I've worked on also in the past. Melanoma, thyroid cancer, also leukemia, which is not on this table, and other types of cancers. These are the solid type of cancers we're focusing on. But this is just the microgravity model. The space radiation risk, which I didn't include the table here, but that's in the paper, is, you know, includes a lot longer list of cancers, which again would include breast cancer, lung cancer, melanoma. You know, would also include, as I said, leukemias for the space radiation, not microgravity models. And lymphoma, and there's other, you know, and then we already know, and NASA recognizes that there's other types of cancer risks out there, you know, in addition to what's here, but it's not as well studied. So we know that, but as you see in those lists, and same in the space radiation list, ovarian cancer or any of these gynecological cancers that are, you know, cervical, vaginal, uterine, and so on, they're not appear in this list because people just haven't studied them. Which is a shame, because we know if there's a risk of all these other cancers with microgravity and space radiation, why won't there be a risk for gynecological cancers, and why isn't it studied, and why can't we understand the impact of that? What's interesting about these other studies is, you know, they always use gamma as a reference because that's always what we want to go to compare to what we know on Earth and in the clinic, and the damage caused by the heavy ions, you know, compared to the gamma, is much higher, and the biology is much higher. And the mutations that form, for example, from, you know, the heavy ions is different. And there are standard, you know, universal damage caused by space radiation, like DNA damage activity, you know, that this is physical activity in the DNA breaks. That's regardless of the type of radiation, but heavy ions will cause a lot more of that. So, you know, there'd be more chance of mutations and so on. So the biology changes a lot with heavy ions. It needs to be understood well. So now, you know, what kind of studies are done on effects of space flight, simulated gravity, and gynecological tissues? This is what's known, and there's not that much papers out here. As I said, you know, in the overall list of papers that are done for the different radiation, there's quite a bit, but here is far and few in between. And you can see it's most of it's like in vitro studies they've done. And what, the effects they've seen, for example, in ovarian, this is from simulated microgravity. There's not very much space radiation as you see here, experiments. Space radiation is here, sorry, on the right table, and you can see not very much happened. But for, let's say, microgravity, for example, they saw different suppression in ovarian cells where, where they've done it, all these are mostly in vitro experiments, but they identify, signatures in these cells, whether they're looking at specifics, genes or doing omic studies, which it's not omic studies, usually specific genes. They see that specific signatures related to ovarian cancer might pop up. They don't see the cells transform, but they see that happen. And same with like cervical and endometrial cancer. Now space radiation, here's the doses that use, and you can see, for example, they've done maybe carbon ions. You see carbon here. X-ray is not very equivalent to as I said, the biology is very different, even though some people might have used X-rays. And then here's some high LET radiation that was used with, again, with single ions. So you can see that, sure, maybe carbon, and it's very sparse to the amount of data that's out there in type of radiation. A lot of these are low LET radiation, as you can see, or protons with a lower energy. So the real equivalence of space radiation, that is well characterized for some cancers. That's not for the gynecologicals. It's not very well understood. But what they do see, for example, is increased DNA damage. Well, we know that's going to occur regardless of whether there's high or low LET radiation. Decreased cell survival. But and things like that can contribute to cancer, which then you extrapolate, as I said, to the amount of radiation damage that could be caused by the heavy ions. So now we drew some hypothesis and correlation to what can occur based on what we know in the literature, from the lower tier, low LET type damage to high LET. Now this is where this is our hypothesis generated from this paper to now further do the work we're doing now to hopefully in the next few months we'll finish the project and we could actually then submit a paper for review. And the idea is that space radiation being a lot more damaging than the low LET radiation, the gamma, what it's going to do based on what we know, what's done already with low LET radiation, that it could deplete like ovarian follicles, and then this would of course increase apoptosis and DNA damage and increase oxidative damage in the female reproductive system, and all of this stuff would then contribute to potential risk of cancer and developing like ovarian type tumors. Here's some other things with space flight. In general, meaning the combination of space radiation and microgravity that could be detrimental for cancer, we talked about, and also just the microgravity component by itself. I mean, there's things that could happen that based on what we know from the previous literature, which needs to be further investigated, is that, for example, ovarian secondary follicle development is disrupted, an abnormal meiotic spindle formation, which that's a key signature of cancer formation. So all this stuff can then eventually lead to cancer, which would be detrimental. So there's also other studies that I just want to quickly talk about that is led by Begum, who's in the RAWG, that a bunch of AWG members are part of, but she's also looking at female reproductive system impact, mainly estrogen and insulin signaling that's going to be changed, which again can lead to cancer. And there's some interesting results there, which I won't get into today. So I hope this was enough to convince you that there should be further work and further research done on gynecological cancers. Join our AWG, because this next project is really looking at the data now, that's available and dissecting that apart. And I thank you for your time, and thanks for everyone to be involved with this project. Thank you.
Okay, so it's actually the time has come to declare the close of this Gynocare training school. It was 3 days packed with knowledge, with a very good social program. And I must admit now I'm being confused whether it's gene or gin or jeans or jeans. But it's been really great. I mean, this is the first training school in this coast section. And thanks to Professor Gligor and his team, I think it has been a huge success. We all agree on that. So big applause to Professor Gligor. And I don't know if you want to have the honor to actually say the last word for this conference.
I need. They put it is. Say a few words Macedonian. First for all the audience and then I will translate it in English. Значи им благодарам на сите учесници, на сите колеги, на сите експерти, на целиот тим што работеше на овој проект. Дојде неочекувано, меѓутоа имавме 3 дена, како што рече Жин, полни со многу знаење, со многу интересни теми. Некои не допираат лично во работата, некои се интересни да ги знаеме. Меѓутоа ова е нешто што веројатно кога си одиме после дома и сега кога ќе заврши, ќе видиме дека всушност ова е иднината на медицината во мало собрана во Скопје денеска во Македонија. So dear colleagues and friends and participants and experts and part of the Macedonian team preparing this practical training school on pathology, on rare gynecological cancers, actually I think that it's a wonderful opportunity, and every day when I was going home, and I think everybody, and especially today after these 3 very tiring days for us with a lot of gin, and I think that it's going home, I always get the perspectives actually that we are actually listening and learning what is the future of medicine. So the things we are listening here, for some of us, they are like distant future. They are, in some of the cases, very impossible. Some of these projects to be done in our countries, but nevertheless, when we are together, we are stronger. So thank you, Jean, for proposing this training school to be in Skopje in Macedonia, and I hope that we will set these standards every meeting to be better and better and better and better, that everybody will enjoy. And we have, as we had here, we had like 40 lecturers from more than 30 countries, and some pre-recorded, some online, some in person, everybody expert in his field, and I think that's a wonderful opportunity. Thank you once more, and I will officially say it that this training school in Skopje, North Macedonia, on rare gynecological cancers, I will say that it's closed. But in the same time, just remember today is Gynecological Oncology Awareness Day, so go out and talk and discuss that with your friends, family, patients, and do some preventive strategies. Thank you.
Thank you. Okay, so now I will end the Zoom. So no longer recording. Yes. So